Hello, everyone, and welcome to our online lecture series for genetics. Today, we embark on probably one of the most important topics in the entire class. One of the things that you can be expected to know, whether you're taking this class here at West Shore Community College, or at Harvard, or Stanford, or the University College of London, or any other school in the world when taking a genetics class. That topic, of course, being three-point test crosses and the ability to calculate recombination frequencies and genetic distances between loci. So, with that grand introduction to the topic, let's see how this builds on our discussion from last time, where we discussed topics of assortment, where we looked at the math surrounding where loci and their respective alleles, or genes in the respective alleles, line up in order to create even distributions across the various gametes that are produced. Now, while we focused on how these chromosomes line up, there's, of course, the topic that we ignored last time, the concept of recombination. The fact that, sure, you can have these alleles lining up on the metaphase plate, attached to different chromosomes, and those chromosomes can determine where they go, but those chromosomes can also be rearranged, forcing us to examine a greater amount of genetic diversity than simply what can be created by chromosomes lining up on sides of the metaphase plate differently. The best way to illustrate this is if you have 23 chromosomes, you can mix and match them, that creates some number of combinations. But if you have something like 20,000 genes, and you can mix and match sets across those 20,000, you can truly understand, or at least begin to appreciate, the amount of genetic diversity that can be created by recombination rather than simply independent assortment along the metaphase plate. Now we have this illustration from an early 1900s paper showing how we think crossing over may occur. That you have these chromosomes. Again, at this point, we wouldn't have really understood much about chromosomes and DNA, but we can see that the patterns will mix and match between the black and the white. Another way to understand this fundamental mixing and matching between the black and the white segments is that there is some sort of inherent linkage between alleles of different genes because of the strand of DNA that they're on. For example, the gene here, and the gene here were previously linked, or rather those alleles were previously linked because they were found on the same chromosome. Linkage is what we use to describe this physical connection by being joined on a single strand of DNA. It was actually discovered when we performed dihybrid test crosses and did not yield our expected 1 to 1 to 1 to 1 ratio. The big clue that we're going to be looking for repeatedly today is which of the classes are overrepresented and which of the classes are underrepresented. Let's make sure we got some of the very important vocabulary and can visualize it, because a lot of this makes more sense when you can see it with your mind's eye. So first and foremost, you may see recombination or recombination rate represented as the Greek letter rho. That's a lowercase p here. Loci that are in linkage equilibrium are loci for which you can see a equal ratio produced in their offspring or in their gametes. So for our locus from last lecture, where we were looking at capital R lowercase r, capital G lowercase g, this set of loci are said to be in linkage equilibrium if you have an equal proportion of gametes produced with all 4 possible genotypes. So that's linkage equilibrium. Now you won't hear that term used a lot because instead when we talk about recombination, we're looking for loci in linkage disequilibrium. What this means, if we copy our text from above, if you have a 1/2 chance of selecting capital R and a 1/2 chance of selecting lowercase r, you have no information about what the probabilities are for selecting the G allele. It's not 1/2 for each, as if the loci were in linkage equilibrium. It's instead a function of how far apart they are. Linkage disequilibrium is the non-random association of alleles. Random association means you have the same chance of getting that allele paired, the big R paired with either G. Linkage disequilibrium is where that association is not 1/4. Now, the reason why we have linkage disequilibrium is because genes and their respective alleles exist on a haplotype. A haplotype is the string of specific alleles found on a piece of DNA. Now you can have 2 different conformations of these alleles. The alleles can be found cis, meaning that in our case, the capital R and capital G are found on one strand, and the lowercase r and lowercase g are found on the other. Or the alleles can be found in trans, meaning that the capital R is paired with the lowercase g, and the lowercase r is paired with the capital G. This helps establish some of our chromosomes and which alleles are found in linkage disequilibrium with one another. The 2 on the same strand are in disequilibrium. The 2 on opposite strands are not in linkage. Now we'll visualize these positions on chromosomes in 2 different ways. A chromosome map, which can also be referred to as a physical map, because it reports things in terms of base pairs. It's going to report the distance between genes in terms of the number of bases that exist between them. But the number of bases is not equal across the genome. Instead, when we look at the genetic map, which is a linkage map, the linkage map is going to report distances in so-called map units, or less commonly, centimorgans, but we don't like to immortalize individuals. Individuals don't matter. Scientific concepts do. That's at least my outlook on things. Centimorgans, or map units as I'll be referring to them in this class, measure the frequency of recombination between 2 loci. meaning that you can have two loci that are 1,000 bases apart on chromosome 1 and two loci that are 1,000 bases apart on chromosome 2, and they can have a different distance in terms of their genetic map. But their chromosome map, their physical map, is the same. One way in which we visualize this and can try to understand the order of genes, which is a problem that we'll be solving in this class. You certainly have to be able to figure out the order of genes given different genetic maps or distances between genes. Let's take a look back at our three loci of R, G, and A. Here we're ignoring any capital lowercase, we're just calling them by their gene names. And you are told that the distance between R and G is 5 map units, and the distance between R and A is 3 map units. Now you can arrange these in two different formations. You can have R next to G with a 5 map unit distance, and G next to A with a distance of 3 map units. Or you can have R 5 map units from G, but A in between the two of them, meaning that the distance from, I did this backwards, R and A would be 3, and the distance from A to G would be 2. This creates two different maps. You have to have more information in order to determine gene order. The way that we're going to be determining gene order is with recombination frequencies, the number of progeny that carry these alleles, and how they've been passed down through meiosis. So, let's take a look at a little bit simpler situation and revisit our chi-squared for two loci recombination problems. Now, if you need a full refresher on this, the complete lectures associated with the topic in Bio 2 will be posted to the course campus page. But we're going to assume that you've had some background on this and see if we can zip through this quite quickly. Because 3 is greater than 2, and we've got to get through 3 today. So first up, you are given a situation where the parents of this individual were true breeding. The two loci we're studying are loci A and loci B, and they each have two different alleles, a 1 allele and a 0 allele. Now, because these parents were true breeding, you can deduce that the haplotypes in this individual are A1 linked to B1, because that was the only combination that this individual could have derived from mom, and A0 linked to B0, assuming that that's the only combination derived from dad. So the parental types here are A1B1 or A0B0. Now, if recombination were to occur in this individual, we could see two different types, wherein A1, let's make a smaller little adjustment here, where A1 connects with B0 or A0 connects with B1. Those give us our two recombinant types. I would encourage you to pause the lecture frequently throughout this section, so that way you can make sure that you understand the topics that we're going through. Lots of pausing, lots of independent deduction here. Now, if we want to calculate the number of map units between A and B, or the genetic distance between A and B, we can take the number of recombinant progeny and divide them by the total offspring and then multiply by 100, or correct them for percentages. So let's say hypothetically that we had a total number of or rather we had 60 parental types split into our A1, B1, A0, B0, 30 each. And then we had 40 of our A1, B0, A0, A0, B1 type in terms of genotyping the sperm of this individual or the eggs of this individual. Now, with 100 total and 40 recombinant, we can tell that the map distance between these two loci in this situation would be 40 map units. Now, keep in mind, what's the maximum distance that two loci can have? Well, the maximum distance, practically speaking, is that they can be found on two separate chromosomes. If they're found on two separate chromosomes, what's the likelihood that mom's 1 gets paired with mom's 2? Well, there's a 50% chance. So if you have two loci on separate chromosomes, you're going to see, in theory, a calculation of 50 map units between them. The fact that we see 40 map units in this scenario suggests that these two are actually found on the same piece of DNA and that they are relatively far apart. If they were 10 more map units apart, they're equally far as though they were on two separate chromosomes. Kind of a cool but important observation to make. Now, whenever we perform chi-squared, or the judgment of whether the map units are correct, or whether these loci are actually on separate chromosomes, chi-squared is our statistical way of testing that, we need to establish a null hypothesis. Our null hypothesis is that the loci are unlinked, meaning that they're found at 50 centimorgans or on different chromosomes, same thing, and that they are in linkage equilibrium. Now, if those parents were true breeding, and these loci are at linkage equilibrium, we can start doing some math in order to determine whether the observed number of sperm or eggs, matches the expected number of sperm or eggs in terms of parental and recombinant haplotypes. So let us set up ourselves a practice problem. Let's say that we have 500 progeny, 500 babies created, 500 egg genotypes. And when you're looking at these 500 egg genotypes, you're expecting them to be equally distributed within these 4 different types of gametes that can be produced. Meaning that you're expecting 500 times 1/4, or 125 A1B1, 125 A1B0, 125 A0B0, and 125 A0B1. Now, you'll notice that I've colored these based on blue here being recombinant, and red being our parental types. I do find that color coding can help with this. We're going to do some color coding later as well. Obviously, it doesn't matter which colors, but just having that visual representation can be useful. Now, we're going to want to calculate how many once we calculate how many are expected, we're going to want to see how many are observed. Now keep in mind, the parental types are actually one class. You do not have an A1, B1 without also creating an egg that is A0, B0. You do not have an A1, B0 recombinant sperm without also creating an A0B1 recombinant sperm. So here we can report some numbers, and now I'm going to change these very, very slightly from what we see in our expecteds, so that way we can reject that, rather accept this null hypothesis, showing that these are in pretty much linkage equilibrium. We have 126 of the A1B1, 124 of the A1B0, 126 of the other parental, and 124 of the other recombinant. Now we can lump these two together to see that we are seeing 252 parental types and 248 recombinant types. So we can go ahead and plug this into the equation. We're going to need to sum across two different classes with our observed number of parentals being 252 minus the 250 that we'd expect to observe. Remember, you're adding these two values together, squaring that, and then dividing the value by 250 before adding it to our recombinant chi-squared statistic, where you have 248 minus 250 squared value divided by 250. Now both of these are 2 squared divided by 250, which actually is 0.16 if I've entered this correctly into the calculator. 0.16 plus 0.16 is going to give you your final chi-squared statistic of 0.32. So, with our chi-squared statistic, we can go to our chi-squared table and see what is the probability of having seen this distribution of observed values if our linkage equilibrium model stating that the loci are unlinked is correct. With a value of 0.32 and 1 degree of freedom. 1 degree of freedom because we have only one class that varies. We see that going down, 0.32 is above 0.6, it's above 0.15, and it's below 0.46. So it's somewhere in this range. Let's go ahead and circle that in a black box. That means that we're somewhere between 50 to 70% probability of deriving these values, assuming that these are perfectly linked loci, perfectly unlinked loci. Perfect linkage would mean that you never see A1 with B0, and we see it with similar frequency as though they were on separate chromosomes. So there we have it, a quick recap of how we perform our chi-squared statistics. Now, for a lot of our practice problems, problem sets that you'll be asked to do, you're going to be given different map units and have to calculate different expected ratios. Keep in mind, the expected ratio was used to derive that value of total expected. So the map unit here was 50, meaning that, or 50 map units, meaning that half of them were distributed into each of the recombinant types. You may have a different ratio here if you were given map units of 20 map units between A and B. Instead of 0.25, this would be 0.1 and 0.1 to get this expected cluster in our recombinant types, and it would be 0.4 and 0.4 for our parental types. Always make sure your brain is on when you're doing this. Make sure that you are not just trying to follow a set formula, but combining numbers that make sense. So ultimately, This is our way of visualizing when we have recombination between two different loci. We can illustrate the parental types and recombinant types and do chi-squared between them to see if those map distances are accurate. But what happens when we take on our next task? 3 loci recombination, or 3-point test cross. Well, here we see our parental types. A1, B1, C1 is one parental haplotype, and A0, B0, C0 is one parental haplotype. This is what things look like if there's no recombination. You can get a recombination event between loci A and B. If there's a recombination between loci A and B, you'll have A1, B0, C0. I should change colors here, sorry. A1, B0, C0. Or A0, B1, C1. This means that you inherit these two together along with the allele from the other chromosome if there's a swap in between A and B. How about if a swap occurs between B and C? Well, in this case, your type would be A1, B1, C0. And conversely, you should see the same thing for our other side. I'm going to draw it in and then erase it because I actually think it's a little bit more confusing now that I'm presenting it. You'll see one chromosomal type where you can mix and match the alleles, but two are always inherited together. Now, we can also have a double crossover. This would be indicated or occur because you have an A1 crossover to the B0 and then crossover again to the C1. This will mean that you look like this parental type at A and C, but not at B. The only way that you can get B in between A and C and have the A and C parental types together is in the event of a double crossover. Once you figure out what the gene order is, it's easy to figure out what the double crossovers are. Conversely, once you figure out what the double crossovers are, it can also be fairly simple to figure out the gene order. So rather than writing these out in these schematic haplotypes, I want to show you these because this is what your brain should be seeing when you write out the following genotypes. So these genotypes for our parental are A1, B1, C1, A0, B0, C0. For our single crossover event between A and B, we see A1, B0, C0, and A0, B1, C1. For our single crossover, this time between B and C, we see the genotypes A1, B1, C0, A0, B0, C1. And last but not least, our double crossover, where we have A1, B0, C1, and A0, B1, C0. Now, if we are looking at the number of sperm created from meiosis in an individual that carries those true breeding haplotypes from their parents, right? We had parents being true breeding, but this time with C1 added and C0 added, what can we determine about the frequencies? Which of these haplotypes do we expect to occur in higher quantities than the others? Well, since we think that these are linked to one another, the parental types are likely to be the highest abundance. You will find the most sperm with the parental types. The single crossover events, where you only need one recombination event to occur between either loci, are our two middle tiers, where you will actually get more recombinants between the loci that are farther apart. So you're going to see more sperm created with more of the A1, B0, C0 type if the distance between A and B is greater than the distance between B and C. And then we will always get the lowest. Always the lowest in terms of the number of double crossovers. This is incredibly important to understand. I know that I might be going over this a couple different ways, but I want to make sure that you see it in a number of different ways. Next up, we'll be looking at individual loci to see if we can identify haplotypes as either parental, make this a little bit bigger here, as either parental or recombinant. When we're given the full genotype, we have to identify whether the relationship between A and B is parental or whether it's recombinant. So let's do some examples. Well, looking at A1, B1 for A to B, we see that both of them are 1s, so we can indicate that this is a parental haplotype between loci A and B. Same is true for A0, B0, also parental. How about between B and C? B1, C1, B0, C0. Parental, 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 parental. Now, in looking between A1 and B0, we see that this is a recombinant type. We also see the same being true for our other recombinant type. Looking at B to C, we see B0, C0, meaning that this is a parental at both of those. Looking at A to C, we see that this is actually recombinant because we see A1, C0 and A0, C1. Take a moment here and fill out the rest. Did you get what I got? Where you should see 4 parental and 4 recombinant types in the relationships between each of our 2 loci at a time. Now, combining our relationship here with this matrix and our expected number of sperm, we can take a look at our next exercise in terms of the expected counts of different sperm produced given a specific map unit distance between these loci. So, let's assume that we have 100 progeny, or 100 sperm. The reason why I use progeny and sperm interchangeably is because if you perform a test cross against a A0, B0, C0, and you look at the genotypes of the babies, since one parent can only donate one haplotype, by looking at the genotype of the individual, you automatically know the other haplotype. If my statement didn't make sense to you, please take some time to try to draw out what a test cross would look like. If it still doesn't make sense to you, let's have a conversation. Now, looking at our toy example here for our expected counts, I will assign a distance of 10 map units between A and B, 20 map units between B and C, and therefore, since map units are somewhat linear, we can assume something like 30 map units between A and C. Now, what are our expected progeny counts, or sperm counts, in each of these categories? Well, first things first, I'm just going to go ahead and copy this table from above. Note that this is all listed in the same way. Actually, I should preface this next part with the qualifier that this actually does not work mathematically. What I'm going to discuss here ends up with numbers that are technically wrong. But this is a useful way to visualize the ballpark of how we can assign numbers of sperm based on these map unit distances. To calculate a true value would be a little bit more complicated, and it's not actually useful to do the correct math here. So I instead am showing you a problem without variance, without any kind of genetic interference. We'll talk about that in a moment. And with numbers that don't actually give you the exact map units that you were given, but it does set you up to see the approximate ratios. So this goes to your sanity checks of making sure that you are not completely making up wrong numbers. So, if we have 100 individuals, we need to figure out how many of our individuals are going to be recombinants between each of our 3 pairs of loci. Now, because 100 and map units are in the same scale, they're both out of 100, both in percentages, the distance between A and B is going to be the number of progeny that we expect. 10 sperm with recombinant AB haplotypes. 20 sperm for B and C, and roughly 30 sperm for our A and C. A and C being the greatest distance here. Now, in order to figure out how many are found in each of our respective cohorts, our double crossover, our single crossover. Sorry, let's go ahead and write this out. Double crossover, type 1, single crossover, type 2, single crossover, type 1, and our non-recombinant type. We can start with the math for the double crossover first. To find our double crossover expected number of sperm, we can take the distance between your first two loci, multiply it by the distance between the next two loci, and then multiply that by the sample size with the distance of 0.1 times distance of 0.2, or recombination frequencies of 10%, 20%, and a sample size of 100, giving us a number of double crossovers equal to 2, meaning that we'd expect something like 2 across this group, meaning 1 and 1 of these. Now keep in mind, we're dealing with small numbers here, but it just goes to show you in this toy example that with small distances between these loci, you won't expect a lot of double recombinants, a lot of double crossovers. For our other two classes, our single crossovers, we will expect to see, again, the distance between the loci times the sample size, or SS, minus the number of double crossovers. So in the case of our recombinants between B and C, where B and C were 20 centimorgans apart, 0.2 times 100, so we'd expect to see 20 of them here, but we have 2 double crossovers, meaning that as a class, we're seeing 18 here, meaning something like 9 and 9. Same equation applies for our other single crossover, 0.1 times 100 minus 0.1 times 100 minus our 2 present, meaning that you would see 8 across this class with 4 and 4 present here. For our non-recombinant types, our parental types, we're looking at the remainder, or the distance between the 1 minus the distance between the loci. Where the total distance is 0.3, so we're looking at 0.7 times 100, or 70. Now keep in mind, when you add these back together, I want to highlight this, showing, really convincing you that this is not mathematically correct. 70 plus 8 plus 18 plus 2 gives you 98. So it doesn't return your total sample size, but it does give you a ballpark of how many individuals you're expecting in each of these classes. Again, I wanted to illustrate this to you so that way you have a sanity check, something that you can see in terms of how these frequencies will be distributed. 70 would be distributed 35 and 35 here, by the way. In terms of the rankings here, we see that there's the most non-recombinants. There's the second most recombinant type that has the largest distance. Third most is our single recombinant type with our smaller distance. And our last and lowest amount is of our double recombinants. So let's take this to another problem. See if we can calculate distances given haplotypes or haplotype counts. So now dealing with our real row. Let's go ahead and copy down our matrix here. I do find this quite useful in terms of making sure that we're assigning haplotypes correctly, except for when we cut off some of the lettering. Oops. That'll do. For this problem here, I will give you the total numbers. Again, I will not incorporate variance, so this will not be a realistic problem, but it'll be a problem for which you should be able to calculate the recombination distances. So in this no variance problem, we see 284 of each of our two parental types. We see 44 of each of our recombinants between A and B. We see 79 of each of our recombinants between B and C. And we see 12 of our recombinants that are double crossovers. Now, just like before, where we want to calculate our map units, where map units are the number of recombinants divided by the total, this is where this matrix is extremely handy. So we first need to calculate our total. Oops. If we add all of these up, we see a total of 838. I think that's correct. Please do make sure to double check my math. For our recombinant types, we're going to take the 44 here, 44 here, 12 here, and 12 here, and add them together. 44 plus 44 plus 12 plus 12 should be 112 divided by our 838. That yields us a recombination frequency of 13.4 map units. So from this equation, or rather from this data, by taking up all of the recombinants between A and B, we can calculate a map unit distance, a genetic distance between A and B of 13.4 map units. Go ahead and take a second and do the math for B and C and A and C. Hopefully you pause the video. I'm not going to wait. Pause is a much more efficient way, so I'll do the math here. We've got 79 plus 79 plus 12 plus 12 plus 12 gives us 182 divided by 838, yielding 21.7 map units. And last but not least, 44 plus 44 plus 79 plus 79, yielding our highest distance, 246 out of 838, giving a map distance of roughly 29 map units. So if we were to go back and grab our chromosomes here, we can see that our distances between A and B were 13.4, 21.7 between B and C, and then overall, this distance is a little bit off. 29 map units. This goes back to the idea that linkage, or our genetic map, is not exactly linear. Mostly linear. In many cases it is genetically additive, where you can add map unit distances to one another to get the ratios. or the relative distances between them, but it's not perfectly additive. And this is because of the last topic that we're going to talk about today, the concept of interference. When you have a recombination event, I'm going to go ahead and copy this over here to try to illustrate my point. And this will further illustrate why we had to go over some of our earlier topics. When you have a recombination event between B and C, let's say that it happens here. There's going to be a lot of machinery that's going to be recruited around B in order to make this happen. A lot of proteins. For that recombination event, you now have actually blocked any recombination from happening in this window, therefore altering the distance between B and C for a subset of recombination events. This is the process known as interference. Now, there are some pretty cool studies trying to better understand this, things that vary this recombination interference, whether it be physical processes or genetic processes in terms of properties of the DNA. But we do get variation in the total number of double crossover events due to this phenomenon of interference. So how do we try to quantify that interference? Well, we do so by looking at interference and the coefficient of coincidence, or the likelihood of double crossover events blocking one another, right? So we're again going to turn to our lovely table, lovely little matrix. Whoop. Sweet, we didn't cut off any pieces there. And we're going to be given different numbers this time. In this situation, we will have variance, meaning that you're not going to have similar numbers distributed across all of these categories. But I'm still going to keep things arranged for you in this pattern that's well known. Now keep in mind, I will not be giving you things in this structured pattern where you already know gene order or parental types. You are going to have to rank them yourselves and fill in this matrix yourself when performing these problems. So, baby steps. Let's first do this question with variance and calculate a coefficient of coincidence and interference. So in this case, we have 244 of A1, B1, C1, 236 of A0, B0, C0, 152, 148 of our two single crossover events between A and B, 89 and 81 of our single crossover events between B and C, and then 23 and 27 of our double crossover events in terms of sperm counts. So you can take a calculator, pause the video, see if you can calculate the map units between each of these. Hopefully you paused. If not, just do it in real time with me having done the calculations for you, but hopefully you actually did them. Please, please, please actually do them. We have 1,000 progeny altogether. Nice even number here. We see that the number of recombinants between A and B is 350. The number of recombinants between B and C is 220. And the number of recombinants between A and C is 470, giving you map units of 35, 22, and 47. In this case, generally additive. Now, In order to calculate our interference, we need to calculate the observed double crossovers versus the expected double crossovers. The observed double crossovers are 23 plus 27, right? We observed 23 plus 27, meaning that we have 50 observed double crossovers. Our expected double crossovers are the distances, again, times each other, so the distance between all three loci, times the sample size, or 0.35 times 0.22 times 1,000, giving you the value of 77. Now there's an intermediate step here. The coefficient of coincidence is calculated by taking the observed double crossovers divided by our expected double crossovers, meaning that we have 50 divided by 77, or something like 0.65. In order to calculate our interference from our coefficient of coincidence, we can take 1 minus the coefficient of coincidence, or an interference of 0.35. And now you can take these measurements of interference and compare them across different species and across different regions of chromosomes, and see how interference and recombination varies across the genome. Kind of cool, huh? So that's where we'll call it a wrap for our, that's where we'll call it a wrap for our notability time. I'm going to end the lecture by transitioning over to Excel for a moment here and showing you how I would set up this exact problem in Excel. This class is highly computational, meaning that there are large portions of this class where you are expected to be fluent with doing math and biology on a computer. So giving you this food for thought of how to set up an Excel spreadsheet to solve these problems might help you out when approaching your own practice problem sets. I'll see y'all over on my computer in just a sec. Here we are with our Excel spreadsheet meant to solve the same problem. Simply because of the way Excel works, I'm coding things slightly differently. A and B and C have changed to 1, 2, and 3, so we have distances between 1 and 2, and 2 and 3, and 1 to 3. We still indicate our parental and recombinants the same way, and just like before, I've color-coded things. We indicate our haplotypes, A's, 1s and 0s as 1s are A's, B's are 0s. That's, again, because Excel would simplify 3 zeros down to a 0 unless we changed the form of the cell, which I'm lazy, so you just got to work within the data frame that you're working. So we have all of our types arranged here, double recombinants at the bottom. We have a total sample size, and then we have our variable count information. So in order to find the total sample size, we can take the sum of our counts. In order to take the number of recombinants of each type, we have to sum across where we see our purple cells. So we could do something a little bit more fancy where if this column equals R, add column B, but you can also do this quite simply. It's a little bit easier for the coding, and I want to keep things easy for this first introduction. We see that there are 350 recombinants between A and B, or 1 to 2, 220 between B and C, or 2 and 3, and then 470 between A and C. We get our map units by dividing the number of recombinants divided by the total sample size for each of these. We got our expected number of crossover events by taking the distance between the first two and the second two loci, multiplying by the sample size. That gives you a number there. Keep in mind, in this example, it's a neat round number, but you might get a fractional expected double crossover number. Our observed double crossover number will always be a whole number, an integer, simply because you're adding together physical counts of sperm or progeny that you're observing. And then from there, we can calculate a coefficient of coincidence, dividing our observed by our expected number of double crossovers, 0.649, rounded up to 0.65, and then our interference of 1 minus the coefficient of coincidence. Now, why is this Excel spreadsheet useful? Well, imagine if you can change some of these numbers. You could then set yourself up some practice problems and see if you can calculate these ratios and calculate this interference. I would strongly recommend you do this because while this problem is fairly straightforward because you're setting things up correctly, by building this spreadsheet and practicing in this way, you build yourself the habit necessary in order to do this on your own when I've scrambled these and not established an order here. We'll take a look at that kind of problem set in our next lecture.